The next question is, does the leech have a pair of paramedial stripes on the dorsal and ventral surface? So I have photographs here of the dorsal surface of our leech and of the ventral surface. And I have used arrows to mark the paramedial stripes that are clearly visible on both the dorsal side and the ventral side. And the arrows that you look at would be the arrows closest to the head region. The second, our next characteristic is, does the leech have 6 pair of crop ceca? The long, dark region that you see running through the center of the leech is the leech intestine. And the crop ceca are the little sac-like structures that extend off of the ceca. So does it have 6 pairs of crop ceca? I have used the arrows on the ventral surface, on the lower surface of the ventral photograph, to indicate 6 structures that I would classify as crop ceca. But Glospinia also have what are called 4 pair of simple ceca located near the posterior sucker that are characteristic of all Glospinia, and the large arrow on the right side of the ventral photograph indicates that area. But my question that I would ask Dr. Govodich is, this first arrow that, near the head that I have marked as a crop ceca, is that really a crop ceca?
Yes, actually, looking at this, the one that you have labeled as the 4 pairs of simple cecae, that's actually what we call the post cecae. So those are still considered ceca. So looking at this, and that first pair is also a pair of cecae. So it looks like you have 7 based on that, which I guess is really one of the questions that you had.
Yeah. Right.
So yes.
So. Yes, so that is a question, and we've classified this as Glospinia complanata, but if it has 7 pairs of ceca, then it may not be Glospinia complanata. And again, this is due to the ability to use the microscope with much better magnification than we had this summer. But I will continue with classification. But the last question is, are the gonopores separated by 2 annuli? Many leech are what are referred to as hermaphroditic. They have both male and female characteristics, and the gonopores are the external openings to the male and female sex organs. So I've used a little black line to mark what an annuli is. The annuli are the exterior segments that are visible in the photograph, and I want you to know this photograph is at 760 times magnification, so that these gonopores are extremely small. So I've indicated 2 annuli for you in the lower left-hand corner of the photograph. And then the 2 arrows represent what I think are gonopores. The male gonopore is much larger. That would be that kind of obscure little area on the, marked with the arrow on the left. And then the female gonopore would be marked with the arrow on the right. And those 2 little areas are separated by 2 annuli. And so I would ask Dr. Govodich or Dr. Bain, are these really gonopores? Or are they?
Yeah, it looks like, obviously it's a little bit fuzzy, but yes, those look like they're in the right position, and yeah, they look good.
So, assuming for the time being that we have 6 pair of crop ceca, we would classify this leech as Glospinia complanata. So, now I'll go to our second leech, and I'm not going to go through all the taxonomic keys. I've kind of shortened it for the sake of time. So first, the presence of a pharyngeal proboscis places it into order Rhinopedella, and I have used a black line to mark where you can see the retracted pharyngeal proboscis. The body is flattened dorsoventrally and much wider than the head of this leech. And as shown in the photo on the right, this leech, again, it has 3 pair of eyes, placing it into family Glosponidae. So the posterior sucker is without a circle of glands, even though I'm not showing it. It has a firm body, and as indicated in the previous slide, the eyes are in 2 paramedial rows, and it has a pair of paramedial stripes on the dorsal surface and on the ventral surface, again indicating that it could be Glospinia complanata. But then when I come to, again, trying to identify the 6 pair of crop ceca, when I count them, I also come up with 7 pair of Cropseca, if I'm counting them right. And that would make it a new species, possibly Borobadella vercata. So I've used arrows on the ventral ones to indicate what I'm questioning as whether those are 7 pair of Cropseca. And I would ask Dr. Gavidich, have I identified those correctly?
Yeah, so again, it looks like you have the 7, including the post-Seekie. So yeah, looks like you got the 7 there.
Yeah. So now, it's nice to be able to do this on Ivan and ask Dr. Gavidich these questions. Otherwise, I have to email him the slides and ask, can you tell me what I'm looking at? And then he has to email me back, and it takes quite a lot of time. If we were connected to a cyber infrastructure network, then he would be able to look at the microscope at the same time that we're looking at the microscope, and he would be able to tell us how to move the slide around, what to focus in on the leech, in order to get the proper identification. So Bordetella, like Glaspinia, has 2 gonopores are separated by the 2 annuli. And I have looked on this leech, and I was not able to identify anything that I would call a gonopore. So again, having the aid of Dr. Bain or Dr. Gavidich would really help us in trying to find these gonopores. So just as an example of how complex these leeches can be, this is an example of 3 other leeches that we have identified currently as Glaspinia campanata. And as you can see in the slide on the lower left, the larger leech is about 1 centimeter, and then that little tiny red blob is another leech that we have identified, and I have shown a magnification of that on the upper left photograph. And I've used a little arrow to kind of mark what we could identify as 1 Sika. So again, a very huge difference in these leeches that we're looking at that we're trying to identify. The other interesting thing about these leeches are that the material in the Cropseca is red, which indicates that it's probably blood. And this is something that Dr. Bain observed when she was with us in the summer of 2010. She actually observed a G complementa attached to another leech, and actually we watched as the Cropseca filled up with the blood from this leech. So again, questions that we would like to have Dr. Gavidich and Dr. Bain direct participation in identifying these leads. So in conclusion, the cyber infrastructure access would allow Dr. Gavidich and Dr. Bain to directly participate in the identification of these leeches and help us confirm what we are identifying and continue with our research. So I would like to acknowledge Dr. Fred Gavidich and Dr. Bonnie Bain from the Southern Utah University, Dr. Kristen Darlin from the University of North Dakota, Dr. Scott Hansen from Turtle Mountain Community College, who is the director of our tribal colleges and university program, TCUP, Dr. Carol Davis, who is our EPSCoR coordinator, Tylen Fountain, who hopefully will be here in the next couple of minutes, an EPSCoR student, Jenny Houle, our TCUP student, and then all the TCUP and EPSCoR students in the summer of 2009 and 2010 who have been willing to get up at 6 o'clock in the morning and help us collect leech, and also the University of North Dakota for allowing us the use of their microscope. Thank you. Are there any questions?